Using the light spot assay, the behavior of a larvae can be tracked before, during, and after entering the light area, allowing the detailed larva movement to be analyzed. This assay is simple and easy to perform. The light response is tested only one time, so that any possible effect of light adaptation can be excluded.
To prepare the agar plates, slowly and evenly cover the bottom of a 15 cm petri dish with an approximately 4 mm thick layer of hot 1% agar. Allow the plate to cool at room temperature until the agar has solidified. To set up the visual stimulation system, clip a prepared LED light source onto an iron frame, taking care that the light projects down toward the desktop. Tilt the cylinder slightly until the angle between the cylinder plane and the vertical plane is about 10 degrees. Connect the 470 nanometer blue LED light to the LED 1 plug of the high-power LED driver and turn on the driver. Turn the knob in the upper right corner of the driver to select channel 470 nanometers and click LED. When the screen displays a check, a blue light spot will appear on the desktop. Click OK and turn the knob to adjust the intensity of the light. Move the position of the light source up and down to adjust the diameter of the light spot to 2 centimeters. Rotate the knob to select the light intensity according to the experimental requirements. Use a compact power meter console with a standard photodiode power sensor to measure the maximal and minimal light power in the spot. The light power needs to be measured in the dark in the actual experiment. Then record the light power, measure it 3 times,